Hi guys, welcome back to part 2 of our macromolecules, and today we'll be going over proteins and nucleic acids. So last time was carbohydrates and lipids, and we're going to hit on the other two today. And just a forewarning, I try to do these recordings when my kids are not here. They are here today, and I have threatened them with their lives to be quiet. Not really, but I have a 13-year-old girl and an 11-year-old boy, and I'm just hoping that by the time I'm done and get down there that all chaos has not broken loose. But anyway, if you hear some loud noises, it's them, and I apologize. Okay, so again, to keep these under an hour, you can read through this on your own, and then the question of the day to be thinking about that we'll come back to at the end is what holds proteins together? So on the pre-lecture assignment, again, this should be done before you watch this, so I'm not going to fill in all of the blanks for you, but I do want to go over a couple important things that I think you need to know before we get started. So if we look down here at this model, we have the structure of an amino acid, and this is one of the monomers that make up proteins. So with these, we have an amino group, and amino just means nitrogen, containing nitrogen. We have an acid group, hence the name amino acid. And then you have the rest of the molecule, which we call the R group. And all the R groups on the 20 different amino acids are going to look different, and that's what causes the variation in these amino acids is this R group right here. Still trying to learn how to run this thing. Okay, so proteins are long macromolecules made from amino acid subunits. I know I said I wasn't going to go over the pre-lecture part, but like I said, I do want to hit on this. There are 20 different amino acids, and each has a different R group. Some R groups are polar. So if you remember the water molecule, when we talked about water, how it's polar, it has that partial positive charge and partial negative charge. And others can be nonpolar. And they can be hydrophilic or hydrophobic. So remember, hydrophilic is water-loving, and hydrophobic is water-fearing. And that will be important here in a little bit. We look at this reaction here. We have two amino acids, so you have the amino group on each one, and then you have the acid group on each one. And we build these together to make proteins using that dehydration reaction that we talked about last time, where we lose that water molecule here, and then that frees up a space for them to bond together. So the dehydration reaction is the building reaction, where we lose that water molecule here. And we call this bond here a peptide bond that bonds the two amino acids together. So now we had our monomers, our amino acids, and through a dehydration reaction, they form, at this point, it's a dipeptide. Di means two. We just have two amino acids. And so down here, that's kind of summing it up. A dehydration reaction joins two amino acids by forming a peptide bond between them. And now we have here a polypeptide, and in this case a dipeptide, because there are two amino acids. Alright, so this next one, protein shape, there's a little activity, I guess you could call it an activity, a little demonstration that I will do at the end once we learn how all of this bonding works. So again, we'll come back to that. We'll come back to the question of the day. So we're going to skip this page for now. Let's see, where do I want to put this? Sorry, I have so many papers here. I gotta shuffle them all around. Alright, so starting with proteins, levels of organization, we have different structures. So primary structure, that's the first structure. Secondary stands for the second. Then we have tertiary and quaternary. So we're going to go over each of these and what they look like and how they together build proteins. So the primary structure is just the sequence or order of the amino acids. So if we look at this here, this is not what a protein will look like. That's just kind of breaking it down to show you. Each of these dots represents an amino acid. So you have this long chain of amino acids, and they're bonded together with those peptide bonds that we talked about. And that is the primary structure, just a chain of amino acids bonded together. The secondary structure is when you get the hydrogen bonding between these amino acids. So you're going to end up with either an alpha, that's the Greek symbol for alpha, an alpha helix, which is what you're seeing right here. So it's kind of like this, what are we calling it? Twisted spiral staircase. Or you get a beta sheet. So there's the Greek symbol for beta. And this is what a beta sheet looks like here. So it's kind of folded like a fan. So if we zoom in a little bit on these, you can see with the secondary structure, there's the amino acids are starting to bond together with hydrogen bonds. So you can see there is a hydrogen bond here, H2O. There is a hydrogen, I tell you, I cannot say that word, bond here. And so they'll start kind of wrapping up in certain shapes because of those hydrogen bonds. With the beta pleated sheet, kind of imagine that chain of amino acids going like this and folding up like that, and then they're held together like this with the hydrogen bonds. Okay, so primary, just the chain of amino acids. Secondary is when they start to bond together with those hydrogen bonds into either that alpha helix or the beta sheet here. And we're going to come back to this question after we go over the other two structures. The tertiary structure is when the protein folds into its three-dimensional structure. And it does this when the R groups form bonds with one another. So if we look over here at our tertiary structure, you can look at it and kind of pick out a few things. You've got a beta sheet here. Got some alpha helixes in here. Helices? What's the plural? I'm not sure. But what they will do is the R groups will start to bond together and fold on top of each other to get this 3D shape. And I think we cover why this happens in just a minute. But basically, we talked about how R groups can be polar or nonpolar. So the polar are going to be attracted to each other, and then, or the ones with charges, will be attracted to each other, and they'll start to fold in on each other. And then the hydrophobic ones are going to want to hide in the middle away from the water. And I'm going to model this with that demonstration in just a minute. So hopefully that will make more sense. And then finally we have the quaternary structure. And this is multiple of these tertiary structures starting to combine. So these guys sticking together. Here I don't have a color copy, and again I apologize because it makes it easier to see, but we have 4 of those tertiary structures bonded together to get the quaternary structure. And this is the structure in large multi-subunit proteins. Okay, so let's come back up to this question here. Make sure you have that written down. My 7th graders always yell at me for going too fast. But again, this is a video. You can always pause it and rewind if you need to, I suppose. So this question here in this box, what would happen to the secondary and tertiary structures under extreme heat or extreme acidic and basic conditions? So what happens is the shape of a protein is everything. The shape of the protein is what allows it to do its job. And if you mess with the shape, it can no longer function. So if the proteins are exposed to extremely high temperatures, like if your fever gets too high, if you've ever wondered why your parents start to get worried if you get a high fever that they can't cool down, I don't know the exact temperature, but getting up to like 103, 104, we're starting to worry. Or if it becomes too acidic or basic, it will start to break the hydrogen bonds in the R groups and basically unravel the protein. So let's define that here. These conditions, the high heat or change in pH, break bonds, holding the secondary and tertiary, structures together, so the structures and the proteins start to unravel. Which will leave them unable to do their function. And there is a big word for this that I mentioned in the last lecture, but it's called denaturation. So again, if your proteins are exposed to extreme heat, or extreme acidic or basic conditions, they'll start to unravel or denature. And once they do that, you can't fix them. You can't wrap them back up. They're just done. They're done for. So you may be thinking like, well, in your stomach, you have proteins, you have enzymes, and that's an extremely acidic condition. I think it's got a pH of almost 1 with a hydrochloric acid. And so how come that doesn't denature? Enzymes are very picky as far as their environments. So that enzyme and those enzymes in the stomach are, I guess, made for those acidic conditions, and if you were to make it more basic, then they could denature. So keep in mind that just because it's an acidic condition doesn't mean the proteins are going to denature. They're kind of specific to their environments. Alright, so let's look at an example of a protein, very important protein in the human body. This is human biology, called hemoglobin. This is a protein found in your red blood cells, and it has the very important function of transferring oxygen to cells throughout the body. So you breathe in oxygen, your red blood cells pick it up from the lungs, it's carried on the hemoglobin and delivered to the cells of your body. And without getting that oxygen, we know that our cells will die. And this hemoglobin protein is made up of 4 tertiary subunits, kind of like we saw on the other page. And an example of what can happen if anything goes wrong in the formation of these proteins. Here is normal hemoglobin. So we've got our primary structure, it's the chain of amino acids, and then eventually we get to the quaternary structure where it's all folded up. And again, this is a normal hemoglobin protein, and this allows the red blood cells to be the shape they are supposed to be, which is kind of like a little bowl. And over here, we have an abnormal protein. So again, mine's not in color, but I think there's a pink amino acid right in here somewhere. And this just shows what happens if you change even one amino acid in the primary structure. And it's all about mutations, which we're not getting into yet, but just a little heads up. If you change just one amino acid in that structure, it can change the shape of the entire protein. So you can see we've got different shapes here. And that makes a misshapen red blood cell, and it's called a sickle cell. And this is where sickle cell anemia comes from. It's just one messed up amino acid here. Changes the shape of the protein, makes for not correctly formed red blood cells. And so it's got the sickle shape, and it can cause a lot of problems. This cannot flow through the capillaries as smoothly as this can, so I should re-study this, but I think it's things like blood clots can cause blood clots, and like poor oxygen delivery to the cells because these misshapen cells can't get to where they need to be. So primary sequence of amino acids, very important. Just mess one thing up and you've got a whole host of problems that result from that. Alright, so down here we've got a whole bunch of molecules, a whole bunch of amino acids. You are not expected to memorize or know any of these. I just want you guys to see that they all start off with the amino group and the acid group. They all look the same at the top here. And again, what makes them different or varied is their R group. So the things that are shaded in or highlighted, bottom parts, they're all different. And that's where we get our 20 different amino acids. So some of them have non-polar R groups, like I said, and these are going to be the hydrophobic ones that are not water-loving. They're going to want to get away from water. And then you have your polar side chains. These ones are hydrophilic. They're water-loving because they can be attracted to the polar charges in the water molecules. And then you can even have electrically charged side chains. And it's not showing the charges on here, I don't think. But at least it's grouping the amino acids into what the properties of their R groups. Alright, so we'll come back to proteins again in a minute. But we're going to move on to our fourth and final macromolecule, which is nucleic acid. And nucleic acid is very important when it comes to genetics. And so we'll discuss this in more detail when we get to that in our, forget which chapter it is, but we'll get there. But a nucleic acid is a macromolecule or a polymer made from nucleotide monomers. Its function is to store information. Specifically, genetic information. So essentially, it makes up your DNA. So again, the monomers, the nucleotide monomers, are made up of 3 parts. You've got a phosphate, a sugar, and a nitrogenous base. So here's a nucleotide, here's the phosphate, here's the nitrogenous base, and then here's the sugar. And just to keep in mind, to remember how these bond, a way to think of it is everyone loves sugar, at least I do, and so everyone wants a little piece of the sugar, so they're all going to attach to the sugar. That's how you know that the sugar is kind of in the middle there. And then over here, we have half a strand of DNA. So you've got the sugar phosphate backbone is what we call it. And then you've got the nitrogenous bases that are kind of sticking out the side. And DNA is double-stranded, and so the second strand that would come here, sorry, I'm out of the camera, would mirror this. And again, we'll learn that in detail in genetics. But you can see each of the monomers here, each of the nucleic acids that make up polymer DNA. So sugar and phosphates linked together. They're linked together through a dehydration reaction. Again, I'll say it a million times, dehydration is the building reaction where we lose a water molecule that clears up space for those to bond together. And the nitrogenous bases here that stick out are red as the code for your genetic material. So let's talk about DNA. DNA stands for Deoxyribonucleic, Deoxyribonucleic Acid, so DNA. And my daughter's spying on me right now. One of them's spying on me. Anyway, when she was about 3 years old, she could barely talk, and I made her memorize what DNA stood for. So I have a cute little video of her. I say, what does DNA stand for? And she says, Deoxyribonucleic acid, and it's so cute. And so I make my 7th graders memorize it as well, and I'm like, if my 3-year-old can do it, you guys can do it too. So you guys can do it as well, deoxyribonucleic acid. And that helps us to see what it's made out of. So the sugar that makes up DNA is deoxyribose. that you see right here. And then, again, this is for long-term storage. Of information. And we call these your genes. As far as the structure, we call it a double helix, and you will have 2 strands of DNA held together by hydrogen bonds. So here is the twisted double helix, and here are some of the nitrogenous bases held together with these hydrogen bonds. If you remember, hydrogen bonds are relatively weak bonds, and so when DNA replication occurs, these need to be split apart. Don't worry about that yet. I'm probably going into too much detail, but 2 strands held together by hydrogen bonds. There are 4 bases: adenine, thymine, guanine, and cytosine. Don't panic when you see all those words. You don't need to know the actual names, but you do need to know that the 4 bases are A, T, G, and C. So just need to know the letters. So the question over here, what is a gene? That's what DNA makes up our genes. It's a piece of DNA that codes for something. Basically, it codes for a trait. Alright, another thing we need to cover is RNA. Another type of nucleic acid. And this has many functions within the cell. We're going to cover these in genetics again, along with the DNA. So in this one, the sugar is called ribose. So RNA stands for ribonucleic acid. It's single-stranded, unlike DNA that is double-stranded. So over here we have a picture of it. You still have the sugar phosphate backbone, but the sugar is ribose instead of deoxyribose. And then you've got the nitrogenous bases, but they're a little bit different. So with RNA, you have adenine, uracil, cytosine, and guanine. So the one that's different here is the uracil. So again, you just need to know the letters and know that the difference between DNA and RNA is that this is a U instead of a T. So we've got the picture of uracil, nitrogenous base over here, found in RNA only. Alright, one of my favorite molecules, ATP, adenosine triphosphate, ATP. So this is a nucleotide, and it's the energy currency of the cell. That's important. And this is kind of what it looks like, just a simple model. You've got an adenosine molecule, and then triphosphate means 3 phosphates attached like this. And we haven't really covered this yet, but anytime you break a bond, it releases energy. And some bonds release more energy when they break than others. And if you think of this bond, it's kind of like, just think of it as a spring, and this third phosphate is just ready to pop off and release all this energy. And that is energy that your cell can use. Energy currency of the cell. So, I think a professor used this example once. Let's see if I can not mess this up. But you guys probably don't get paychecks anymore. I did when I was in college. So you would work, you would get your paycheck. Say you got a $500 paycheck from your job, and you take that $500 paycheck to Walmart and try to buy groceries. They're not going to accept that. You have to go to the bank first to get it to the right currency that you need, which is cash. And then you can take that cash to Walmart, and that's a form of money that Walmart can accept. So, for example, when you eat food, it's going to be broken down into glucose, which is our short-term fast energy. And that glucose is going to be, long story short, and we'll learn about this in cellular respiration. It's going to be converted into ATP, which is the form of energy that your cells can use. And the donation of this third phosphate and the energy that comes with it can change the shape of proteins. And just to show you how nerdy I am, I don't even know if I can get this to show under the Elmo, but, let's see. Okay. Here's my cringy tattoo, but it's the cellular respiration reaction. It shows how we get the formation of ATP. So yeah, when we get to that, I'll show this to you. And then when my students get tested on this, I have to cover this up. On the other side, I've got the reverse reaction of photosynthesis, but yeah. Pretty nerdy. That's how much I love science. That's how much I love this stuff. Okay, down here, we have a couple of examples. So here's an enzyme called lactase, which breaks apart lactose. And in order to do this, this takes movement of molecules. It's going to break down lactase into glucose and galactose, and all this takes energy. So it's going to use ATP to do this. Over here, this is so tiny, let's see. We have, cells need to get things in and out, and we'll learn more about that on our next lecture in cells. But sometimes these particles have to be pumped against a concentration gradient, and that takes energy because that's not the direction they would naturally flow. So your body is using ATP all the time. It's the reason you have to eat. So your body can do cellular respiration and turn that glucose into ATP energy that your cell can use. Okay, we are now back to here. Sorry again, I've got papers all over the place. Let me find my stuff. Alright, so protein shape, and we went over this already. They're long chains of amino acids folded into three-dimensional shapes. So we're going to do a little protein folding activity. You can do this at home if you'd like, or you can just watch me. It says using wire, make a protein with 6 to 8 amino acids. Now, this is a bachelorette pad here. I don't have wire. I mean, maybe I have floral wire, but I borrowed some wiki sticks from my classroom at school. So a wiki stick is just, I think it's just yarn covered in wax. But anyway, it gets the job done. So you can either use wire, whatever you have. This is what I'm going to do. So it says, it's kind of like we're making the primary structure of the protein, just the chain of amino acids, and they all look a little bit different. And you need to have at least one positively charged loop, which is a long closed loop, kind of like this one. And you need to have at least one negatively charged loop, which is a short closed loop like this one. And then we have our hydrophobic loops like this, and you should have more hydrophobic than the positive and negative charges. And says to put about 3 fingers between each of your amino acids. So anyway, if you're doing this at home, just pause and go ahead and form something that looks like this with 6 to 8 amino acids and do a mixture of these 3. And what we're going to see here is how it folds together. So you've got the primary structure here, just a sequence of amino acids. And then I can't really model the secondary on this, but just imagine that it's either coiled in some way or in that beta sheet folded kind of like a fan. But then we want to see how this would fold together to make the tertiary structure. So we know that these here are hydrophobic. They're afraid of water. They're water fearing. And so they're going to want to orient themselves away from the water. So most of your proteins are in water. So as this starts to form its tertiary shape, all of these hydrophobic parts are going to want to fold in towards the center. And this isn't working so well because it's kind of sticky, so I don't know how good of a 3D shape we're going to get. But those will want to go toward the center. This is just my leftover end here. And then your positively charged and negatively charged loops will start to attract to each other. So maybe these two will fold over like this and bond together. And then this negative and positive charge will fold over like this and bond together. And now you've got this 3D crazy looking protein structure. That actually turned out pretty good. Now, if you were in the classroom in person, I would have you guys match up with 4 other people and stick your 4 together to make a quaternary structure, but at least here you can see the tertiary structure. All right, so let's fill in what we need to fill in here. So question of the day, what holds proteins together? So with the primary structure, those amino acids are held together with peptide bonds. And then that secondary structure is held together with those hydrogen bonds. Matt, do you need something, bud? Matt?
Am I allowed to talk?
Yeah, just real quick. I'm still recording now.
How long are you going to be?
Probably 10 more minutes.
Okay.
And then when we get to our tertiary structure, when I was folding everything in like you saw, we have ionic bonds. So that was the positively charged attracted to the negatively charged. And we also had water pushing away these hydrophobic bonds to the center. All right, so why is your shape different from your neighbor's? Well, if you can imagine, if we were all sitting in class and made these, we wouldn't have chosen the same sequence of amino acids. I mean, maybe by random chance. but they would fold differently on each other because they have different amounts of these hydrophobic groups or different amounts of the ionic bonds forming between those twisted loops. So basically it's different because they would have had a different sequence of amino acids. And then the last thing we need to fill in here is protein shape. So again, the protein shape determines the function of the protein. So shape is everything when it comes to proteins. And the linear sequence of amino acids determines the shape and therefore the function of the protein. Hey Matt, it's too loud, thank you. Sorry. Okay, so that is it for our proteins and amino acids. It's the end of our macromolecules, but I do want to show you guys something that may be helpful, and that is just kind of compiling all of this into one chart. So if you want to pause this and draw this outline, and we'll fill it in together, and then if you can like redraw this on your own without looking at your notes or anything, these are the basics that you need to know for the exam. So we've got our polymers or our macromolecules. So we've got carbohydrates, lipids or fats, proteins and nucleic acids, and you need to know the monomers, the building blocks of each. So for carbohydrates, the building blocks are saccharides or sugars. In lipids or fats, you've got the 3 fatty acids and the glycerol molecule. The monomers for proteins are amino acids that we just saw. And the monomer or building blocks for nucleic acids are those nucleotides. Alright, so make sure you can do that. Name the monomer, the building blocks that make up each of these, and then know the function in the body. So the carbohydrates, the monosaccharides, the simple sugars are for fast energy. So the example for this was like glucose. Once it's in your blood, your body's going to use that first for energy. And then we have our polysaccharides or our disaccharides, which are 2 monosaccharides together, and then polysaccharides, which are many saccharides together. And these are both like intermediate forms of energy because you have to break bonds through those hydrolysis reactions in order to get it to a monosaccharide so it can get into your cell. So it can still happen pretty quick, but not as fast as the monosaccharides because they are already just ready to go. The function of lipids or fats, this is your long-term energy storage. So for energy uses, again, your body's going to go to the monosaccharides first, then to the di and polysaccharides, and then if it still needs more energy, it'll break into the lipids, the long-term energy storage. So the functions of proteins was a long list. I don't think I'm going to fill it all in, but you can. But things like support, enzymes, transport, defense, hormones, motion in your muscles. I don't even want to write just one because I don't want you to think it does one thing. So I'll just write many, and then you guys can review your notes to see what those are. And then the nucleic acids, like your DNA and RNA, they store genetic information. So my advice would be for you to redraw this chart like I had it first, and maybe just do these top three so you have the 4 polymers memorized as well, and then just fill it in and have this memorized. And then, this is a little easier, I think, but know the difference between DNA and RNA. Sorry for my crooked lines here. But DNA, remember, for number of strands, it's double-stranded, while RNA only has 1 strand, single-stranded. And the type of sugar in DNA is deoxyribose. So remember D and D, not the game or whatever it is. I don't play video games. The type of sugar in RNA is ribose. So remember the R and R. I do like R and R. That stands for rest and relaxation, right? Not something bad, hopefully. Don't know what I'm talking about. Okay, and then the bases in DNA, we have A, T, G, and C. In RNA, the bases are the same except for the T, which is substituted with a U. So we have A, U, G, and C. So again, I would practice drawing this chart, filling it in, and that will help you be prepared for the exam. And that's it for macromolecules, and we'll see you next with cells, one of my favorite things. Have a good one.